So we start with the demonstration of the new MES QwikCheck DFI Kit. This is going to be your low melting agarose. What you need to do first is to melt this low melting agarose. So you can either put it in a beaker full of water and put it into a microwave, or you could melt it at 80 degrees with a dry bath. The choice is yours. But when you melt it, it should come down as droplets. Right now you tap it and it's solid.
Yep.
And when you melt it, it's supposed to be into droplets. And then you place it at 37 degrees Celsius. So while it's sitting in the 37 degree and trying to be as molten as possible, you can put that one also inside. Yeah, you can dilute the sample. The sample should be diluted. This concentration was estimated to be 60 billion. Yeah. So according to this, you should be diluting it to 1:6. So you'll be taking approximately 100 microliter of the sample and 500 microliter of the diluent. Yep. You now need to take 40 microliters of the diluted sample and add it to the 60 microliter of the low melting agarose. Can you take that out? You can always ensure that it is molten and the temperature is compatible by placing it over here, because the temperature would be more indicative and it is still molten. So, and if you delay it any further at the room temperature, it might just get too cold and solidify again. So you want to watch. And it's better to have the slides out at the right time. We are going to make 2 slides. These are pre-coated agarose, pre-coated slides to help the low melting agarose bind on it. Mix the diluted sample well and make sure that you don't bubble it though.
Just mixing it with the agarose.
Now we are going to add 20 microliter of the samples in 2 parts and make sure you have the cover slip ready. I can prepare first. Keep them 2 separate. Okay. So this is how we are going to place the sample droplets. One above the other at the lower end of the slide. And let the sample flow. And you see the sample is flowing on its own. You don't need to actually press it. It will occupy its own space. And now you can place this in the refrigerator for 5 minutes. Just making another one.
Should I put it?
Yeah, you go ahead. You see, if you delay it anymore, it's already solid. So you have to be really fast with it. So in order to make another slide, I just use the diluted sample and make just one more using this one. So it's still under molten stage. To add 40 microliters, mix it well without bubbles. 1 and 2 and 3 color slip flow. You can refrigerate this. You can see the rectangular film forming on its own. The agarose will flow. Okay, put this in the refrigerator for 5 minutes. And meanwhile, while we are waiting for the agarose to solidify, what we can do is we can make the Lyse Plus solution. So the Lyse Plus solution is going to be your Lyse A powder plus your Lyse A solution. You'll be adding 1 ml of the Lyse A solution to the Lyse A powder. Simply add it from the top, close the vial and roll it between your palms, and it's done. I'm going to repeat that. Alternatively, you can use a pipette to mix it, but be sure not to form a lot of bubbles because it tends to be soapy. We'll be waiting for 5 minutes. Fresh out of the fridge, and you make sure there is no additional moisture, which it's not going to be. It's just 5 minutes. All you need to do is place your thumb here, no pressure, you just slide it out, easy, and that's it. Comes off. You just leave it to get accustomed to the temperature. Can you get the second one, please?
30 seconds.
In 30 seconds. So we're going to just allow it. It's okay to refrigerate it a bit more, like 7-8 minutes, 10 minutes. It's fine. It just makes it more solid. But we can't just leave it like overnight or something. So better to remember. So we've got the second slide out, and this is exactly what I'm going to do. Wipe the back side, make sure there's no extra moisture, and this is all I do. You might want to see there is a rectangular gel. You can see the definitions, right? And yes, now we're going to put them on a staining rack and start staining them. So the first thing that we do is add the Lyse A solutions that we have prepared. So we're going to keep the timer for 5 minutes once we add the reagents. We recommend 600 microliters should be enough, but yeah, you're not going to store them, so it's okay to pour it all out. And wait for 5 minutes. And it's always good to ensure that nothing is like falling out and the entire rectangular film where your agarose film was is covered, and you don't leave any place which is like not covered. Yes. you can just like wipe the back side of the slide, and this would be a good time to observe it under microscope to see if we have accomplished having a good lice.
So the kit provides references at every step of the way, so you can double check your progress.
And this is what you're supposed to see. Like you can see that the head, which otherwise would have had all the black DNA and the nucleus, you can actually see that they've gone a bit transparent. Like you don't have anything in them. They're almost empty, or they appear empty. Like you just need to, yes, you can see these. This is how they're supposed to look. They're not supposed to have anything black inside. This would be a perfect example.
Good.
Right. I'm going to just capture this one for reference. Okay. Now it's time to add lice B.
Come in here.
Just tap on Zoom.
Yeah.
Okay. After the lice A, if you're not really busy looking at the vision, the best thing to do would be to add the lice B solution and add it like double quick. And just pause a minute. Make sure you add it to the entire slide, and there is no part which is not having that one. It's simply not this stuff. Make sure to make a bigger hole. I'm just making sure this entire slide is covered with lice B. And you just keep it for like 5 seconds. Make sure the whole slide gets it, and decant. Again, you can observe this step under the microscope, or the vision would be a nice place. So now you see that you can just see the tails. Like you'll really need to focus to find the heads, because they've got really transparent. You can't see the heads anymore. Can see this one? It's gone really empty. These have like really gone empty. So I'm just going to capture this one for reference, and we will add the other reagents. The next thing to add would be the fixative. So again, the fixative, you add it on the whole slide and wait for 2 minutes. Yeah. So once we are done with the 2 minutes of fixative, we can decant of whatever is remaining. It's okay if there is nothing remaining and it evaporates, and all you do is drop the stain. So the stain, you take it for a minute. Just make sure that, yes, just make sure that the stain covers your agarose film. It's really dark blue when you add it.
One note, you're focusing on the front inch or so of the slide, because that's the part that'll get visualized.
You can actually see the film here. If you could stand here,
yeah,
you can see the rectangular film of the agarose. So you'll know exactly where to add your reagents.
Got it.
Just make sure that the entire rectangular region of your agarose film is covered, because your sperms are going to be all over it. So it's better to stand ready with Jimsa B And remember, while adding Gimsa B, you're not going to remove the Gimsa A, which is already there on the slide. So all we are going to do is add Gimsa B right on top of it. Just make sure that it doesn't flow away from the slide, and you will notice there will be a change in the color from blue to purple, and there will be 2 minutes of wait here, over which you will see, just make sure that the entire film is covered this way and it's mixed. And while you're waiting, you can see a metallic shine or sheen coming over, so that shows that it is reacting with your cells. You can see that one, it's something like a layer which petrol forms on water. Can you see that? And here we go. So once you're done, you might be curious as to how it will look. All you have to do is like decant the stain, and you'll find that it looks all purple and no longer blue. So you just wash off the extra stains until you see that no longer anything blue comes out, and you can see it is forming a rectangular mat already. So this is how you wash it, and once you're done, it makes sense to wipe the back side of your slide to remove extra water, never the front of course. And you see it's all purple. So the best made slide should have this rectangular thing very, very visible. And we wait until it dries.
How long usually until it'll dry?
Depends upon the humidity of your place, but I think 5-10 minutes should do it. If you find that you have a darker background and you would like to have a sharper contrast between your background and the halos, all you need to do is a bit of your Gimsa B. Can you see the name? Yeah, Gimsa B. And just swirl it around a bit so that if there is some extra stain, it's going to come out. Gimsa B will make sure that the phosphates on your DNA, they keep the stain for themselves, while only the background will lose its extra stain. You can see the blue ones coming out. A little bit of blue will come out, and then you just pour it out. And then you dry the stain, and then you observe it under the microscope.
Do you recommend they don't try to do anything funny like setting it on a heating pad or anything?
To dry it?
Yeah.
No, because it might
Just
shrink
leave it alone.
everything.
Yeah, yeah.
We don't want it to shrink. Like, do you see the improvement in the contrast?
Yeah. Yeah, it is a little bit.
Yeah. So you can see a decent head, halo definition, along with the tail. So, and if you allow the slide to dry, you can see the tails in all of them. So sometimes you preserve the slides for too long and you think that there are too many cracks on the agarose. No worries, you just have to dip it in the water, like the back side of the slide, and observe it under the microscope or the vision, and you can still see a good contrast. So all of these have tails, unless like you can focus. You can see this one, and I think this one is very good. This is a small halo clearly, and you have the large halo, the medium halo ones, and of course there will be always a sharp contrast between the head and the halo, and we are supposed to measure the distance between the head and the halo.
Expand the image to full screen to make counting easier. First, we want to select the sperm that have no halo or no halo degraded. Next, we want to choose the option to scale, which will help us measure the core and the halo. When selecting cells, first start with the core by clicking on each side of the observation, and then click on the outside of the halo. The SQA Vision software will then tally the size of the halo for you and automatically populate the results on the visualization screen. Moving on to another cell, I'll once again click the core, then allow the system to measure the overall halo by clicking on the ends. Cells with smaller halos will be measured automatically. And you would continue this process for up to 100 or 200 cells total to get a good statistical analysis. The images are saved permanently in the archive and can be brought up at a later time for confirmation or for archiving.